Hi everyone, I'm Claudia, and today I would like to talk you through how to investigate the risk from disease in a proposed assisted colonization of the Sihek, an extinct in the wild species. First of all, I want to thank all the co-authors and partners involved in this project. Conservation translocation is defined by the 2013 IUCN guidelines as the intentional movement and release of a living organism where the primary objective is a conservation benefit, which will usually comprise improving the conservation status of the focus species locally or globally, and or restore natural ecosystem functions or processes. Conservation translocation is an incredibly useful tool for restoring threatened species, and as we can see in this map here, it is widely used across the world. The map shows the percentage of case studies published by the IUCN up to 2013 across the regions of the world. There are different types of conservation translocations. When they occur within the indigenous range, they can be a reinforcement when conspecifics are found within the release site, or a reintroduction when the focal species has disappeared and so is no longer present in its indigenous range. Differently, when they occur outside the indigenous range, we talk about assisted colonization when the aim of the release is to avoid population extinction, or ecological replacement when the aim is to perform an ecological function lost by extinction of an original form. In any conservation translocations, there are different risks, 7 main categories of risk. And the disease risk, which is the focus of this work, is one of these. Let's see now a little bit more in details to whom the disease risk is. An animal should always be seen as a biological package, that is, a host and its parasites. And so in any conservation translocation, when animals are moved, their parasites are moved too. And these may influence the health of the host itself following translocation, or could pose a risk to recipient population or even to the ecosystem as a whole, other species at destination, including domestic animals and humans. It is, however, very important to consider not just those parasites which are moved with the host, but also those parasites at the release site and to which the released animal might be immunologically naive to. The reintroduction of gray wolves to Yellowstone National Park provide a good example of exposure to novel parasites. We saw epidemic mortality in naive wolf cubs due to canine distemper virus, and also population declines in some packs of wolves because of sarcoptic mange. A disease risk analysis is therefore an important evaluation preceding a conservation translocation and can be used to inform any disease risk management, possible pre- and post-release monitoring, and ultimately whether a translocation should proceed. The method we use is published in the 2014 OIE IUCN Guidelines for Wildlife Disease Risk Analysis and involves 6 steps: the description of the translocation pathway, consideration of barriers, hazard identification, disease risk assessment, disease risk management, and disease risk communication. For those of you who don't know much about Sihek, let me quickly introduce this amazing bird. Once common on Guam, it has experienced severe declines following the accidental introduction of predatory brown tree snakes to the island in the 1940s. By 1984, less than 50 Sihek remained in the wild. 29 birds were then captured and transported to US mainland zoos for captive breeding as the last hedge to extinction. The last sighting of a Sihek in the wild occurred in 1988. Currently, the Sihek exists only in conservation facilities under human care, with a population which is estimated at about 135 birds across 25 institutions. I also want to give you an update on where we are at with the Sihek recovery project. We're taking a phased approach. First of all, the selection of the training sites. We have initially identified two sites, Cocos Island and Palmyra Atoll. But unfortunately, we had to drop Cocos, at least for now, because as you might have heard, brown tree snakes have been found on this island too. Palmyra has therefore become our choice, and we're now assessing its appropriateness as a learning training site. Our ultimate goal is to reintroduce Sihek back to Guam, and such learning step will help improving reintroduction success. Introducing Sihek to Palmyra represents an assisted colonization, which is a powerful tool for species recovery where reintroduction is not possible. Yet it raises additional risks to the recipient ecosystems. First step in our disease risk analysis is the consideration of the translocation pathway and evaluation of geographical and ecological barriers. The pathway for this translocation is pictured here, and as you can see, barriers are crossed. Birds will be sourced from mainland ESA institutions and will be taken to their destination sites. The fact that there are barriers crossed here means that there is an increased risk of exposure to novel parasites. And this is because when there are barriers, it means that parasites, either species or strains, may differ between source and destination. And therefore, when we cross these barriers, the risk is increased. Animal populations, which may be naive and therefore lack immunity, might consequently suffer disease. We then identified potential hazards through literature review, both published and grade data, through a survey of captive Sihek holder institutions, and through elicitation of expert opinion. In total, we identified 63 potential hazards, including those known to be present in Sihek or closely related taxa, multi-host parasites, apparent commensal parasites, and also non-infectious agents. We categorize hazards as different types. We have source hazards, which they are those found at the source, but novel at the destination site. Destination hazards are those found at destination, but they are novel to translocated animals. Carrier hazards are those commensal parasites, which may cause disease under stressful situations. Population hazards are those which are present at both source and destination and may have a negative impact at the population level. And finally, transport hazards, which are those which might be encountered during transport and may be novel to translocated animals or the release site, and they're usually moved with equipment like crates, food, and water. In this picture here, you can see again the proposed translocation pathway, but this time I've also written down where the hazards identified by the letter CDPCT, where those hazards might occur along the proposed translocation pathway and where they might cause disease. In an assisted colonization, we are mostly concerned about source hazard, and this is because of the risk of co-introducing potential novel pathogens to destination. However, not all potential hazards might represent a risk. And so we worked with a panel of wildlife health experts to prioritize the hazards. We provided the experts a brief summary on the distribution, the host range, transmission mode, and possible consequences for each hazard, as well as information on the translocation pathway. And we asked their opinion on whether each identified hazard either remains a potentially serious concern or is a minor concern. Most hazards were deemed low risk, but 8 hazards listed in this table here were prioritized as higher risk. Of these, West Nile virus, avipox virus, Themicryozospora species and Trichomonas are categorized as source hazard. We have started to carry out a disease risk assessment for each one of these 8 hazards by producing detailed fact sheets for each one. This fact sheet summarizes the biology, epidemiology, pathogenesis, and also clinical signs of each hazards. So where to next? We are going to quantify the risk by providing the detailed hazard fact sheets to a panel of experts, and we will ask them 3 specific questions. First of all, what is the likelihood of a Sihek carrying and releasing a pathogen at the destination? And in the event of this occurring, what is the likelihood of other species at destination being exposed to and becoming infected with a novel pathogen released by the Sihek? And if this occurs, what is the likelihood of significant biological, environmental, and economic consequences occurring? Quantifying these risks will allow us to decide if it is acceptable or not to introduce Sihek to Palmyra from a disease risk perspective, and we aim at doing so in the next couple of months. As the last thing, if you would like to keep up with what is happening with the Sihek project, then you might like our quarterly newsletter, which covers many different aspects of the project. So here below, you can find a link that you can use to subscribe. Thank you all.